Let's appreciate complement system. A complement system is a group of enzymes present in a normal blood as proenzymes. Their activation constitutes a primary method in which antibodies attack on foreign bodies. They are called complement system because they complement our immune system. There are 20 proteins, but only 11 of them has effect. They are named as factor B, factor C1, factor C2, factor C3, factor C4, factor C5, factor C6, factor C7, factor C8, factor C9, and factor D. But factor C1 has 3 sub-factors: factor C1Q, factor C1R, and factor C1S. These proteins catalyze a series of reactions in which its end product has immune effect. These complement factors are synthesized principally by the liver, but some other ones are synthesized in other organs, like C1 is synthesized in the intestine, C2 and C4 by the macrophage, C5 and C8 by the spleen. What that triggers complement system makes them to follow 3 pathways: the lectin pathway, the alternate pathway, and the classical pathway. The classical pathway is initiated by antibody-antigen reaction. The alternate pathway is initiated by bacteria and toxin, while the lectin pathway is initiated by mannose binding lectin. The classical pathway is due to the reaction of antibody and antigen forming antibody-antigen complex. Antigen-antibody complex. The antibody involved is the immunoglobulin G and immunoglobulin M. The immunoglobulin M is more potent, while immunoglobulin G is has less effect. So the immunoglobulin G is only IgG1, IgG2, and IgG3 that are involved. The binding of the antibody to the antigen causes a conformational change at the Fc portion of the antibody, opening the active site of C1, principally the C1q. This C1q leaves C4 into C4a and C4b. The C4b cleaves C2 into C2a and C2b, C2a and C2b. The C4b and the C2a join together to form C4b2a, and this is C3 convertase because it has the potential of cleaving C3. Cleave C3 into C3b and C3a. The C3 convertase with the C3 forms C5 convertase, OP2A 3P. This is C5 convertase because it has the potential of cleaving C5 into C5A and C5P. The C5B recruits other complement factors: C6, C7, C8, C9, resulting to the formation of membrane attack complex, which we will explain later. In alternate pathway, unlike classical pathway, which requires the presence of antibody, it does not require the presence of any antibody. It is rather triggered by bacterial endotoxin, a lipopolysaccharide present at the cell membrane of gram-negative bacteria, or a yeast wall, or a snake venom. In this case, there is a spontaneous hydrolysis of C3 into soluble C3. Hydrolysis. This hydrolysis causes the opening of the active site of factor B. Factor B is cleaved by factor D into BA and BB. The C3 joins together with BB to form C3 BB. This is C3 converters. It is fluid phase C3 converters. This because it has the potential of cleaving C3 into C3A and C3B. The C3B joins with BB to form C3 converters. This is a stable C3 converters and is stabilized by a properdine. This is C3 converters and this is C3 converters. Though they don't have the same structural formula, they have the same potential. This C3 converters is capable of cleaving C3. So in this place, it follows the classical pathway. This C3BB also has the potential of cleaving further C3 into C3A, C3B, and C3A. The C3 converters together with the C3B forms C3B, BB, C3B. It is 3B. And this is C5 converters because it has the potential of cleaving C5. This is C5 converters. This is C5 converters. They don't have the same structural formula, but they have the same potential. So it can cleave C3. And the rest of the pathway continues as classical pathway. In lectin pathway, it is due to the presence of an organism with either mannose or umbilicus adhesive membrane. The presence of this organism is recognized by membrane-binding lectin produced by the liver. So this is, let's say this is our candida. So injection of this organism is recognized by membrane-binding lectin presence in the liver. So it will bind to it and stimulate other proteins like membrane-binding lectin serine-associated protease 1 and membrane-binding lectin serine protease associated 2. So the presence of this will now triggers the cleavage of C4 into C4A and C4B, and also the cleavage of C2 into C2A and C2B. So this C2B and C2L joins together to form C4A, C4B, 2A. This is C3 converters. It can cleave C3. It's the same thing as this. The membrane attack complex produced at the cell membrane serves as a transmembrane protein, resulting to osmotic imbalance and osmotic lysis of the cell. The C5A produces chemostatic substance, which recruits white blood cells to the site of the injury. The C3B causes opsonization of the cell of the antigen, resulting to phagocytosis. The C3A, C4A, and C5A are called anaphylatoxin as they triggers the mast cell to release its content.